Hey guys, just to put a cap on the Module 2 lab, I know that that's been up for a little while, and I just want to give you some suggestions when it comes to approaching that quiz that's going to come up here on Sunday and Monday coming up. So one of the things I want to do is kind of share my thought process when I go through and do the dilutions and microbial counts. The math is pretty simple, but a lot of times people really struggle with this stuff. So let me just give you my step-by-step process when I'm going to try to solve a problem. And these are the kinds of problems you're going to need to know how to do when it comes to these dilutions and spread plates. It's all about counting and finding out a concentration, right? So we need to understand dilution factors and things like that. So let me give you my steps. Here's the example that is similar to the example we had in the lab, except they had 5 tubes instead of 3 tubes. So the first step I would say is you want to find the original sample. You want to find the original or stock solution, sometimes they'll say, that is the original that you want to find the concentration for. Okay, so here in this problem, they come right out and tell you this is the stock solution over here. All right, so as soon as I find that, I want to label the stock solution, and one way to label it would be 10 to the 0. This has not been diluted at all, and that's what we want to find our concentration of. So first step, find the stock solution or original stock or whatever it is and label it 10 to the 0. This is what we want to find our concentrations for. We want to then label all actions. By that I mean find the arrows. Okay, so one of the actions is they're going to take 1 milliliter here and they're going to put it into a tube with 9 milliliters. Well, I'm going to label that. I'm going to label that as a 1 to 10 dilution. Okay, now let's look at the bottom here. 1 to 10 is the same as 10 to the negative 1. I'm going to use that because eventually my dilution factors are going to be in powers of 10. So I'm going to say that this action is 10 to the negative 1. I'm going to label my next action. The next action is 1 milliliter into 9 milliliters again. Well, that's pretty much the same deal, right? So we're going to have a 10 to the negative 1 again. The next tube, same thing. 10 to the negative 1. Right? As long as we keep things in powers of 10, the math will be simple. Now I see a few more actions. I see some arrows down here where some amount of the solutions in these tubes are then going to be placed onto the auger of these plates down here. And it says 0.1 milliliters will be placed on the surface. Now, 0.1, we find 0.1 over here. Well, 0.1, 0.1, again, is the same as 10 to the negative 1. It's almost like we're doing another dilution here because we're putting just some fraction of a full milliliter. Okay, so this, I would give this action a 10 to the negative 1 label. Every one of these is a 0.1 milliliter, so I'm going to give it again 10 to the negative 1 label there, 10 to the negative 1 label there. Now, I'm done with step 2. I've labeled all of my actions. Now I want to label all of the tube dilution factors. Okay, and at this point, everything's going to be super easy because you've done all of the work ahead of time. What we need to do is consider what was in the stock solution. Okay, let's change colors here for a second. In the stock solution, we had a 10 to the 0. Nothing was diluted yet. Then we have a portion of it taken out and placed into the tube. Well, all we have to do now is add the powers of 10. Okay, 0 plus negative 1 is negative 1. So we have a 10 to the negative 1 inside this tube. That's the dilution inside that tube. Now we're going to do this again. we have an action of 10 to the negative 1. In our second tube, well, we're going to just add again the powers, negative 1 and negative 1, and we get a negative 2. We do it again. We add the powers, 10 to the negative 3. Then we look below and we see what's going to happen in these plates. So in the end, perhaps a problem that might be posed to you is to give me the dilution factors in these 3 plates. I give you everything that you've seen here on the slide, right? But then you have to work out all of these steps, all right? So this last step would be there's 10 to the negative 1 in this tube, number 1, and there's a 10 to the negative 1 action taken when we take just a portion of it and put it on this plate. And so within the plate we're going to have 10 to the negative 2. We do the next one. Okay, we have negative 2 in the tube, we have negative 1 taken out. We're going to add the powers. We have 10 to the negative 3. We do the next one, 10 to the negative 4. Right? So this is the way I would approach these kinds of dilution factor problems. Let's look at the problems that were posed at the end of this laboratory, okay, at the end of the Module 2 lab. So here's number 2 from the Let's Review section, okay? Again, we're going to use all the same steps, okay? So steps are the same. Let's go and figure out what the dilution factors in the tubes are here. They haven't taken us all the way to the plates, so we won't have to do step 4, but we can do all the rest. So stock solution, there it is. I'm going to label it 10 to the 0. I need to label all the actions. I see 3 arrows here, okay? 1 going into 9 milliliters. Well, that's just a 1 to 10 dilution, okay? So we're going to have a 10 to the negative 1 place there. The next step, we have a 0.1 milliliters going into 9.9. All right, this is not quite as simple now. What we need to do is to move the decimal point, okay? And this is almost like we're taking 1 milliliter and putting it into, move the decimal point again, 99 milliliters, okay? Effectively here, we're doing a 1 to 100 dilution. 1 over 100, that's the same as 10 to the negative 2. So this action is 10 to the negative 2. We've diluted it more by the fact that we are taking less solution and adding it to more in this tube. Okay, now we do it again. 0.1 into 9.9. This is again, pretend like you're moving the decimal point in both places. It's like 1 in 100. This is a 10 to the negative 2 dilution. All right, we've done step 2. Now we're on to step 3. We need to label all tube dilution factors. For this, all we have to do is add powers, right? So let's switch colors here for a second. We have a power of 0, a power of negative 1. We add those for the dilution factor in this tube, and it would have to be 10 to the negative 1. 10 to the negative 1 is our answer to part A. All right. Next one, we are going to add powers. It's negative 1 in this first tube. It's a negative 2 action that we've taken. We're going to have 10 to the negative 3. 10 to the negative 3 is our answer for that tube. We do it again. We add powers. We have negative 3 and a negative 2. We have 10 to the negative 5. 10 to the negative 5 is our answer for C. Okay? All right, let's take a look at question number 3 in the Let's Review section. We're again taking the same steps. However, they're actually not showing us the actual dilutions here. They're just giving us 3 tubes and telling us what those dilution factors are already. Those particular dilution factors are already given to us here, here, and here. This is what's in the tubes, okay? So we do not have to do step 1, 2, or 3 in this particular problem, but we do need to label all of the plate dilution factors. That's what A, B, C, and D are. We need to give those, okay? All right, so we need to label some actions here, right? Some of the actions here would be 0.01. All right, that's the same as 1 over 100 or 10 to the negative 2. Next one, we have a 10 to the negative 1 action. Here's another 10 to the negative 1. And here's a 10 to the negative 2. Now we need to add powers, right? So we have a negative 2 in the tube. We have a negative 2 action taken. We have 10 to the negative 4 in this plate. Again, we add powers. Negative 3, negative 1. We have a 10 to the negative 4 in this plate. Same dilution factor in these 2 plates. We just got there in a different way. Next one, we have a negative 4 with a negative 1, 10 to the negative 5. And then we have a negative 4 and a negative 6, 10 to the negative 6. All right? So this is my thought process when I see these problems. Find the stock, give it a 10 to the 0, label any actions, any arrows you find, label dilution factors in the tubes, and then label all of the plate dilution factors using those powers of 10. All right? All right, the last thing I want to address to you guys is the metabolic chart. I suggest that you build a metabolic chart, and I'm fine with you using it for the quiz. In fact, it'll be really helpful during the quiz if you do your homework and put together a metabolic chart, okay? So in this chart, these are the things that I would include. We have several types of media that we learned in this particular laboratory. We have TSA Auger, we have Msa Auger, we have blood auger plates, we have all sorts of media. You want to label the type of media. You want to give the key ingredients of those particular media, the type of media that they are, all the possible observations that you might see when organisms are growing on that plate or tube or whatever. And then you want to enter the corresponding interpretations for each of those observations. So if you see one observation, what does it mean, in other words, for that last column? So let me show you my way that I would go about this, okay? So you're going to have the test, the medium, whatever it is. You're going to have what type is it, contents, observations, interpretation. So let's do one together. Let's do TSA. So TSA Auger, what type of medium is this? This is a complex medium. This is going to include lots of different ingredients. What is contained within a TSA Auger plate? Well, there's a whole lot of stuff there, right? We didn't really go into the details, but we just said that there was a mixture of lipids and carbohydrates and all the things that cells need to survive. So we just write a mixture there. What are we looking for in a TSA plate? Well, we're looking at colony characteristics. Size of the colonies, shape of the colonies, if they're elevated, right, the odor, all of those things. And then we're going to have an interpretation based on that. We're going to write down all of those things. Let's do another one together. Let's do the Msa Auger. Okay? Msa was a selective and differential plate. This was the type of medium we were working with. What does it have in it? It had mannitol, it had salt, and it had a phenol red pH indicator. But there are lots of different things we might see on an MSA plate. We might see that the organism hardly grows at all or doesn't grow at all, so no inhibited growth. We could see just growth where it grew but otherwise nothing happened. And we could also see growth with some kind of color change to a yellow color. So in this last column, what we do is we line up what each of those tell us about the organism. If there's no inhibited growth, we'd say that it's a non-halophile, right? It's non-halophilic. It's not capable of growing in that salt environment. If it grew, but there was no color change, well, then we'd say it's halophile, right? But it is not fermenting the mannitol that happens to be in that plate. And the last thing is we could have growth with a yellow halo. This would indicate that there was a halophilic organism. It's a salt-loving organism, and it's also fermenting the mannitol in that plate. If you put together a chart like this, but not just for these two, but for all the plates and tubes that we work with, and I'd probably even put in the oxidase test and stuff like that, even though they're really not media, right? And they're not selective or differential, but they are useful tests that we need to know about. I would include them all in a chart like this. It's kind of like a cheat sheet. And I'm going to tell you to go ahead and use that cheat sheet during the quiz. All right? So let's say you have this cheat sheet in front of you, and again, I'm just showing you a couple of rows of this chart, and then I give you a picture like this on a quiz. And I tell you this is an MSA. I'd have to tell you a little bit about it. It's an MSA plate. Well, if you look at this, and you first thing you'd notice is that there's no growth at all here. Well, immediately you know the observation is no growth. And you can immediately tell me that this was a non-halophile. This did not grow. It's a non-halophile. You can't say that it didn't ferment mannitol because you didn't give it a chance, right? You killed it with the salt. So this would be your answer for that particular plate. What if I gave you this mannitol salt agar plate? Well, first thing I see here is there's absolutely growth up here, right? So there's growth all over the place. And I also see a yellow color change. So we go to our row that says MSA plate. I see growth here. I see yellow halo. I'm going to select that one, and I'm going to tell you that that's a halophile and a mannitol fermenter because it did indeed change the pH in that plate. All right, so just a few thoughts I wanted to share with you. Let me know if you have trouble with doing any of these problems, and I might give you a couple of microbial count examples that you could play with. They're a little bit harder ones, not as hard as I'd put on the quiz, but it could give you some practice, okay? So let me know if you have any issues or questions about this